Thank you very much, Natalie. It's a great pleasure to be here. So the title of my presentation is Autism Spectrum Disorder, or ASD, Genomes to Outcome. And the messages that I would like you to take from this presentation are threefold. The first is that genes are definitively shown to be involved in autism. The second is that using genomic technologies such as microarrays and DNA sequencing, we can now detect roughly 20% of the genetic forms of autism. And as we move into using higher resolution technologies such as complete genome sequencing, this may in fact move into the 40 to 90% range, and I'll talk about this. Third, autism, and this is most importantly, autism is clinically and genetically extremely heterogeneous. So really like this photograph here, there's lots of different fish. I think this is a grouping of many different disorders, and I'll give you examples throughout my presentation. But the key to taking a genomic approach really is to try to identify very early on individuals that are at risk at autism so that intervention approaches such as applied behavioral therapy approaches and perhaps some medicines can be applied to help improve outcomes for these children as they develop. So what is ASD? This is a heterogeneous spectrum disorder that encompasses deficits in three domains of function. The onset is typically less than 2 years old, although in some cases there are regressive forms where it occurs later than this age. We see deficits in social communication, repetitive stereotypic behaviors, and language deficits. And depending on the extent of these deficits in these three different domains, one would have a different categorization of ASD, including Asperger's disorder, where mainly social communication deficits exist, autistic disorder, child disintegrative disorder, or PDD-NOS, pervasive developmental disorder not otherwise specified. And under the new DSM criteria that will come out in the spring, in fact, all of these different subtypes will be grouped under one category, the DSM-5, which we'll call ASD. So I think this will simplify diagnosis, but in fact probably complicate some of the management implications, and I'll talk about it a little bit later. What's very important is that really we see a very, very diverse effect here. So there are probably many people in the audience, perhaps some anyways, who are Asperger's. In fact, we see this in our faculty. Many professors are on the spectrum, and this seems to increase over time. But what we're really talking about is trying to develop personalized therapies for children that are severely affected by autism. We see medical comorbidities in upwards of 25 to 40% of cases, anxiety or ADHD in perhaps 30 to 50% of cases, although this is not part of the actual ASD diagnosis, and intellectual disability or cognitive deficits in upwards of 40 to 60% of cases, plus other syndromic features that I'll talk about a little bit later. So in fact, in addition, roughly 20% of parents are on the spectrum. This is a subclinical syndrome, which we call the broader autism phenotype, or BAP. There's significant variable clinical presentation within a family, even in identical twins that presumably carry the same mutation. In roughly 90% of cases, they will have very similar clinical outcomes, but in about 10%, you can be on each end of the spectrum, Asperger's and severe autism. So this is really a dilemma in the field, and certainly complicating genetic counseling. There's this interesting phenomena of the 4-to-1 male to female gender bias, and I'll come back to that a little bit later. And now we see in the newspapers every day the statistics that 1 in 88 newborns will receive an ASD diagnosis. So this prevalence seems to be on the rise. Epidemiological data is suggesting this, and the reason why is not yet clear. So pre-2000, we were really living in the dark ages with respect to understanding of why ASD came about. And certainly for idiopathic forms of autism, there were no evidence-based etiologic factors known. So in fact, most of the causation was attributed to how families, parents raised their children, in particular how mothers treated and interacted with their children, the so-called refrigerator mothers syndrome. And this is really an environmental influence. We hear a lot about environmental influences in autism, and I'll come back to this kind of using the capital E environment, which can be things like toxins or, say, more direct effects with behavioral relationships with family members. And I'll talk a little bit near the end of my talk about what I would call the small e contributions. In fact, innate factors or biological factors that may influence onset of autism. But of course, the last decade was wrapped up in the publicity around the MMR vaccination being the primary cause of autism, which in fact has not been shown scientifically to be the case. But the parents are desperate for answers, and they'll look for answers in any place, medical answers or fictional answers, to try to explain what's happening to their child, why it came about, and what they should do about it. In Canada, there's over $3 billion per year spent in services. And again, most of this is not based on having a formal diagnosis or following any type of evidence-based research protocols. So what I'll term the genomic enlightenment, I think really happened in two phases. The first was a study of families, in particular identical twins in the early 1980s, showing that, in fact, genetics is one of the major contributors to ASD development. And the second is using genomic technologies to actually start to find the genetic factors that are underlying ASD risk. So there's been many twin studies performed and studying the concordance rates of ASD in monozygotic twins versus dizygotic twins suggests that genes, in fact, are a major factor. The concordance rates range really from 50 to 90%, depending on the study and depending on the criteria that are used. There was a paper published last year that suggests genetics is less of a contributor, but in fact, if you look closely at the data, it still strongly suggests that at least 35% heritability exists in that study. This drops down to 10% in all of the studies, again suggesting genes are involved. As I'll show you in a moment, we see ASD-related comorbidity in upwards of 10% cases. And through the HapMap era, genome-wide association studies using common variants were completed by multiple groups, including ours in Canada as part of the Autism Genome Project. And the data suggests that for common SNP-type variants, the role is very minor, and it may be additive, but perhaps not a major factor. The real story seems to lie in rare genetic variants. In particular, copy number variants, or CNVs, and single gene mutations that are highly penetrant, suggesting that, as I said earlier, there's rare forms of monogenic in some cases, or perhaps oligogenic type effects in ASD. So the challenges in ASD treatment that we're really trying to have an impact on, the major challenge is diagnosis is quite difficult. The features that you're measuring are features that are part of the developmental process. So language development, for example, you can't really test for up until age 2. And that's too late to have a maximal effect in intervention approaches. So in Toronto, for example, the average age of diagnosis is about 4.5 years. In other areas in Toronto, actually can go up to 5 or 6 years old, depending on where you're living. This is too late to have intervention approaches. There's also the issue, as I said, of many medical comorbidities, things like seizures, intellectual disability, anxiety, and how that affects the treatment protocol. Many families experience this odyssey of diagnosis where they go to multiple clinics. This child shown on the left-hand side of my slide went to 8 different clinics at the Hospital for Sick Children and did not receive the proper diagnosis until a clinical microarray was run. This can lead to expensive visits, expenses of the healthcare system, and in particular, really frustrating experience for the families. So we think that the solution really is to have a clinical, to really explore the clinical and genetic heterogeneity through a genomic or personalized health type approach. And that's what I'll talk about going forward. On this slide, this individual we found in I think it was 2007 carried a chromosome 16 microdeletion. This is the most common form of CNV that we see now, and it affects 0.75% of the Canadian ASD cohort that we've been studying now for several years. Since this Genome Canada funded project actually was completed a few years ago now, the data has been transferred into clinical diagnostic laboratories, which are in the hospitals in Canada. And in the province we're in now, Ontario, there's well over 1,000 clinical microarrays run on primary ASD diagnoses to try to use this in a confirmatory diagnostic way, but also to try to identify ASD earlier on and to get that information to the families when they need it. So this is a summary of what we know of the genetics of autism. On the left-hand side of the slide, ASD-related syndromes. There's over 100 different syndromes where we see ASD associated with it, and the most prominent being Fragile X syndrome. Remember that, I'll come back to this, Rett syndrome, tuberous sclerosis, and others. We know historically through looking at chromosomes, rare chromosome abnormalities can be involved in upwards of 5% of cases or so, the most common being chromosome 15, maternally inherited duplications that we see in 1 to 3%. And more recently, using clinical microarrays and high-resolution research microarrays, we see these copy number variants or deletions and duplications of genes in roughly 5% of cases, and I'll come back to this a little bit later. And more recently, using a combination of microarrays and sequencing or directly doing exome sequencing, other high-risk genes have been identified, like the synaptic scaffolding or shank genes that I'll talk about in a moment. But this adds up to roughly 15 to 20% if you take a naive population of ASD diagnoses and run all these technologies. That's the yield that you get. Suggesting that we need to use higher resolution technologies. There could be multigenic factors involved. Perhaps we need to look in other regions of the genome that are not covered by the experiments we're performing. And we also have to address issues like environmental influences. So when genes are identified, more often than not, but not always, they're identifying genes that are involved in the synaptic complex. So in particular, rare CNVs, and now more specifically point mutations, have been identified in neuronal cell adhesion and structural synaptic complex genes. And as I said, this accounts for roughly 15% of ASD diagnoses. So this is an image of, or a graphic of the synapse, in which, highlighted in red, are the genes and proteins that have been identified now where highly penetrant mutations have been identified. And what I want to point out is that in any given case, there's no single gene that it actually accounts for more than 0.75% of an ASD cohort. And in fact, in most cases, it's less than 0.1%. So these are collections of rare mutations that have a very highly penetrant effect in that individual. That is the risk factor that's involved. This was the first image when we modeled SHANK2 mutations in rat hippocampal cells that was quite striking. I think if you think back to explaining to parents why their child is autistic, showing them an image here of what the synapse complex looks like is really quite striking. And what you see on the left-hand side of the slide is a control experiment, and on the right is when you overexpress this specific mutation in the SHANK2 synaptic scaffolding gene, you see a change in the spine volume along the neuron. And it's probably hard to see here, but it's quite striking when you look at this and measure this in multiple experiments. So we hope that having this new genetic information will actually impact the clinical management of families. And the reason this is threefold: earlier detection of risk for ASD and related conditions, better understanding of the specific causes of autism with implications for medical management and genetic counseling, and targeted individualized approach to ASD treatment. This is the kind of experimental paradigm that we've been following using higher resolution technologies as they become available. We start with blood DNA or saliva DNA, and then typically run them on microarrays. We're running genomic microarrays or gene chips that have 1 million different data points across them, the highest resolution arrays. We've been performing next generation sequencing, exome sequencing, capturing that 1.5% of the genome that codes for proteins, sequencing that, and more recently doing whole genome sequencing. That is decoding the 6 billion chemical bases of information in these individuals and their families. So I'm going to run through 5 examples where we use some of these different technologies and show you the data. This is one interesting one where we've used microarrays to detect a SHANK1 deletion, a very rare deletion found in a Canadian family. The extent of the deletion is shown in the red bar, and it knocks out several exons of the SHANK1 gene. SHANK2 and SHANK3 were already known through our other studies to be involved in autism. And in fact, as Tom Hudson pointed out, when you do these studies, you have to go worldwide because these are very rare mutations. And we didn't find the second mutation in an unrelated individual until we looked through a French cohort. In fact, this individual was from Sweden. And the common region of deletion of overlap shown in red here also knocks out the SHANK1 gene. So these are the first indications. We don't see this deletion in any controls, by the way. And these are de novo, or at least the one on the left is a de novo deletion, and the one on the right is an inherited deletion. So we did lots of sequencing, find other point mutations that further implicate this gene. When we look a little bit closer at the family structure, this is quite interesting. I should point out that SHANK1 is on chromosome 19, so this is not an X-linked gene. I'll come back to that in a moment. This is an autosomal gene. What we see is in the index case, or the proband, the 64 kilobase deletion is, I'll have to go through this a little bit quickly, is only present with an ASD diagnosis, which is indicated with the black box in males that carry this deletion. So female carriers are unaffected. And this is really striking when you look across this deletion, and we have a few other loci identified that I won't talk about today, where only males, probably because of the unique biology and brain development, we think that the gene actually has effect only in males. So carrier females, even though it's an autosomal gene, do not have any ASD-type symptoms. And as I said, we don't see this in well over 10,000 controls. So this is the cause of autism in this family. We went ahead and have been doing whole genome sequencing. I don't have a lot of time to talk about how we go about doing this, but we're using Illumina-based sequencing, where we've now analyzed a few hundred trio families, and I'll just show you the unpublished data throughout the next part of my talk on the analysis of the first 33 of these families, and it's quite striking. To summarize, we found genetic loss of function type variants in roughly 50% of the families. And we think that these changes actually, in many cases, for these 16 families here, will actually have a direct effect on their clinical management, and I'll show you some examples. So we found de novo mutations, which are loss of functioning, all these mutations I talk about, in 6 out of 33 families, or 18%, X-linked or autosomal inherited alterations in 10 out of 33, or 31% of ASD probands. And in some cases, there's combinations of mutations identified. So I'll just talk about these 3 families in the next few slides. So the first is a de novo nonsense mutation in the autosomal caprin-1 gene. The index case of the autistic boy is shown with the black box and the arrow. His age of diagnosis in this case was 7 years old. That's too late for him to actually effectively respond to intensive behavioral type therapy.